Okay, imagine this scenario. An endoscopy, really complex procedure is done. The doctor's just snipped out this tiny bit of tissue, a polyp, right?
Right.
And it could be the key to everything for the patient. But here's the real challenge, the kicker. It's that tiny sample. It needs to get out of the body, travel through all this tubing, and land in the lab perfectly preserved. It's like a logistical high wire act. So today, that's what we're diving into, that critical phase after the polyp's removed. And we're getting our info straight from a great source. Dr. Raju's YouTube video, HCC 1472-Lesson 9C Tissue Sampling Polyp Trap. It really breaks down these steps.
Absolutely. And our mission today really is about that specialized practical know-how, the stuff that turns a successful snip into actual diagnostic information. We're honing in on two main things. First, how you retrieve polyps based on their size, small versus big. And second, maybe even more important, how you handle them correctly so the sample is actually useful for the pathologist, sample integrity.
Right. So we're looking for those maybe surprising practical tricks and tools. The logistics. What actually happens when a polyp is small enough to get sucked up? And, well, what if it's just too big for that suction channel? Let's start with the small one. So a small polyp may be removed with a cold snare. The first thought, the intuitive thing, must be just suction, right? Suck it up through the biopsy channel. Seems fastest.
And that's exactly where you can go wrong if you're not careful. If you just use straight suction, like through the main line,
Well,
that little piece of tissue, which needs to be identified, it just gets sucked right down into the main suction canister.
okay, wait. Why is that so bad? Can't they just fish it out later?
Well, you could, maybe, but it totally messes up the chain of custody, which is crucial. The lab needs to know, with certainty, not just what tissue it is, but which specific polyp it was, from where in the colon it came, and from which patient. Throwing it in the general waste bin, you lose all that specific location context. Diagnosis becomes, well, potentially impossible or unreliable.
Okay, so that makes sense. High-tech procedure meets specialized plumbing almost. How do they actually solve this problem then?
They use something called a polyp trap. Think of it like a special filter attachment. And the procedure requires a really specific adjustment right away. You have to physically disconnect the normal suction tube right where it leaves the endoscope at the outlet. Okay, once that's off, the polyp trap gets connected directly to that scope outlet, and the other end of the trap connects back to the main suction canister line. So now, when you apply suction, the polyp gets pulled into the trap's little chamber first. It's safely caught there before the suction continues on to the main waste container. It's actually quite clever filtering.
Ah, okay, that makes perfect sense, a little detour chamber. But what about efficiency? Say the endoscopist is moving quickly, removes maybe 3 or 4 small polyps one after another.
Right, good point. And that's where the sort of manual procedure detail is key. If multiple polyps are removed, the person assisting has to physically rotate the chambers in the polyp trap system between each sample, if you don't rotate it. The second polyp just lands right on top of the first one. You're back to the same problem. You can't tell them apart. Potential cross-contamination. So rotating ensures polyp A is in chamber 1, polyp B in chamber 2, and so on, nice and separate.
Okay, so this is all very precise. Specialized tubing, filters, sterile handling. But here's the bit from the source video that, honestly, it kind of blew my mind. When it's all done, and you need to get that tiny sample out of the trap for the pathology lab,
Yeah.
the tool they use isn't some fancy, expensive gadget.
No, it's fascinating, isn't it? The key tool for getting that delicate little polyp out of the trap is, it's just a basic toothpick.
A toothpick? Seriously?
Seriously, a simple wooden toothpick. It gives you that perfect sharp point for precision. You gently nudge the polyp out of the trap chamber and carefully place it into the formalin jar for preservation.
Wow. A toothpick. I love that. Simple, cheap item, but used with surgical precision in this incredibly high-tech environment. Human ingenuity right there. All right, so the trap system works great for anything small enough to fit through the suction channel. But let's be real. Sometimes these polyps aren't pebbles. They're more like boulders, relatively speaking.
Yeah.
Too big for the scope's suction channel. What do you do then when suction just isn't an option?
Yeah, exactly. And that brings us to method number 2, the retrieval net. So the polyp's been cut free. Maybe it's just floating there in the colon. The issue isn't just size, it's also slippery, right? And you're working in a pretty confined, dark space. The net is designed to basically grab that larger piece of tissue safely and securely without damaging anything on the way out.
Okay, a net, how does that actually work? Is it tricky to handle inside the scope?
Yeah. Mechanically, it works a bit like other tools you insert, like biopsy forceps or maybe a cytology brush. But the way you handle it, the ergonomics, that's really important. Typically, the operator uses their right hand, Thumb goes in the handle ring, and then the index and middle fingers control the slider mechanism. Gives you very fine control.
Right. So sliding it open pushes the net out into the colon. Sliding it closed pulls the net back into its little protective plastic sheath.
Ah, a sheath. Why the sheath?
That sheath is vital because the net itself, the mesh part, it's made of wire. You don't want that wire scraping against the colon wall when you're inserting or retracting it.
Got it. So you insert the net while it's still closed up inside the plastic sheath. You guide it through the biopsy channel all the way to where the loose polyp is, and then I guess you deploy it. Open the net.
Exactly. You push the slider, the net opens up.
Yeah.
Then you carefully maneuver it to sort of scoop up or trap the polyp.
Sounds a bit like trying to catch a fish in a dark pond with a tiny net. Kind of. And then this is critical. You close the net back into the sheath, capturing the polyp inside, and then you withdraw the whole thing, sometimes along with the endoscope itself.
Requires some serious dexterity, I bet.
Oh, absolutely. And this is where teamwork becomes non-negotiable. The source material, Dr. Raju's video, really emphasizes this. You absolutely cannot operate that net carelessly. The person handling the slider must work in sync with the endoscopist, the one who actually has the camera view. Why? Because when that net opens, the wire frame is exposed. If you're not careful, if you open or close it without clear visual confirmation.
You could snag the colon wall.
Precisely. You could capture and actually rip the delicate lining, the mucosa of the colon. That's a serious complication you want to avoid at all costs. So clear view, careful coordination, it's vital.
Okay, that makes total sense. Safety first, always. Now, let's shift from getting it out to keeping it good. Preservation. Once this big, maybe fragile polyp is out, how do you prepare it for the pathologist? Standard procedure, I assume, is just drop it in a formalin jar.
Yeah, for many cases, that's standard. Retrieve it, put it in the formalin.
But the source pointed out something more specific, right, for certain situations, like when the pathologist needs to be absolutely sure about the edges, the margins.
Spot on. If getting a definite assessment of the resection margins is critical, meaning, did they get all of the potentially problematic tissue right up to the edges, then just dropping it in the jar isn't quite enough.
Why not? What happens?
Well, think about it. You take this three-dimensional, often floppy piece of tissue and drop it in liquid formalin. It tends to curl up, fold onto itself. It loses its original shape and orientation. And that makes it incredibly difficult, sometimes impossible, for the pathologist looking under the microscope to figure out which edge was which. They need to know the true relationship of the tissue to where the cut was made. Was the cut clean, or was abnormal tissue left behind right at the edge?
Okay, so how do you maintain that orientation?
This is another one of those clever, practical solutions. You take the large polyp, lay it out carefully, maybe spread it a bit flat, and then you pin it to a small piece of cardboard.
Pin it, like with actual pins.
Usually, yes, small pins, carefully placed. You're essentially stabilizing its shape, making this 3D sample behave more like a 2D one, preserving its layout. Then the entire cardboard piece, with the polyp pinned to it, is gently placed into the formalin jar.
Ah, I see. The cardboard keeps it flat and oriented during fixation.
Exactly. It preserves that crucial information about the margins, which can directly impact decisions about further treatment for the patient. It's a small step with potentially huge implications.
That simple logistical detail, the cardboard pinning it, really highlights the journey from removal to diagnosis. It's all about giving the lab the best possible information.
And it loops right back to safety, too. Remember that warning about the net?
Ripping the mucosa.
Yeah. The reason that careful, collaborative net operation is so stressed is because a mistake, a rip, creates an immediate problem bleeding, maybe even a perforation, avoidable complications. So careful handling, preserving orientation, avoiding injury, it's all part of the same commitment to patient well-being and accurate diagnosis.
Wow. Okay, we've really unpacked a lot today. We've mapped out those two distinct pathways for getting tissue samples from inside the colon to the lab bench. So now you know about the suction and polyp trap system for the little guys, keeping them identified, and the careful teamwork needed for the retrieval net when dealing with larger specimens.
Yeah, and we've seen it takes this whole range of tools and techniques, doesn't it? From the quite sophisticated, like the multi-chamber trap, down to the surprisingly simple, like the toothpick and the cardboard. Each step, each tool plays a part in turning what the endoscopist removes into something truly useful, something actionable for diagnosis and patient care. Every detail matters.
That sums it up perfectly. And it leaves me, and hopefully you listening, with one last thing to chew on, sort of a reverse perspective. We've spent all this time talking about retrieving the samples. But just think for a second about the tools that do the initial cutting, like that cold snare. Imagine the engineering challenge there, designing those tiny instruments to cut precisely and to ensure the piece they remove is actually suitable for one of these retrieval methods we just discussed. The whole process starts way back with the design of the cutting tool itself. Something to think about.